Um, Now, with my great pleasure, we are having the first talk today from Yvon Jaillais. He's from ENS Lyon from France. And please take it over, Yvon.
Thank you very much for the introduction and for the invitation to speak at this Plant Cell Atlas meeting. I will try to introduce more about lipids, and in particular anionic lipids, and how this could fit into this Plant Cell Atlas initiative. So in my lab, we are interested in anionic phospholipids, mainly because they have two very interesting attributes. The first one, they are membrane lipids, but they are present in very, very low concentration, which make them very good signals within membrane. And the second one is that these lipids, they have the anionic head groups, and this allows them to recruit protein in different membrane of the cell. So you may be familiar with some of these lipids, the very famous phosphoinositides that are involved in canonical signaling also from animal cells. You have also phosphatidic acid. And one of my favorites, and that's the one I will talk today, you may not be familiar with it. It's called PS, or phosphatidylserine. And phosphatidylserine is also an anionic lipid. I think it's very interesting, but not so much is known about this lipid. So the first thing that we did was to analyze the localization or the subcellular distribution of this lipid. And for that, we use so-called sensor strategy, which is actually very simple. We take a protein domain that specifically binds to, in this case, PS. We fuse it with a fluorescent protein, and then we express this fusion in transgenic plants. The idea behind this is that it's going to be expressed in the cytosol, and then it's going to be targeted to different membrane through the interaction with this cognate lipid, in this case, phosphatidylserine. So here I show you a confocal picture that we made in the root of Arabidopsis thaliana. So this is a transgenic line stably expressing this sensor. And you can see that PS accumulate here at the plasma membrane strongly, and also is present in some intracellular compartment that we showed through colocalization to be endosomes. It doesn't really matter for today's talk. So this strategy, this sensor strategy, has been used widely in yeast, animal cell. But one thing that we do when we work with plants is that we work with entire plants, and immediately under the confocal, what we can do is look at the localization of these lipids in different cell types or different differentiation status. And for example, using root, it became very quickly evident that PS is not present at the same concentration in different cells. So for example, in the root epidermis here, what we find is that in the meristem, the vast majority of the PS sensor localized at the plasma membrane. We have very few endosomes, so very strong accumulation of PS in the plasma membrane. And that's not true in the more elongation or differentiation part of the root, where we have a more partition of the sensor with less at the plasma membrane and more into intracellular compartments. So if you want, for us, this was quite interesting because we have specific patterns of PS at the subcellular level within the cell. This was kind of expected, but we also have patterns at the tissue scale. And in this case, we have this gradient of PS with a strong accumulation of at the plasma membrane in the meristem and less in the differentiated part of the root. And for us, well, we started to ask what could be the function of such a variation of PS within the plant cell plasma membrane. So to tackle this question, we took a genetic approach and we analyzed, in particular, one enzyme that's called phosphatidylserine synthase 1, or PSS1. This enzyme is the only enzyme present in the Arabidopsis genome, supposed to make PS, and it's making PS by exchanging the head group of phosphatidylcholine, so exchanging the choline head group with the serine to make a phosphatidylserine. So this is a very abundant structural lipid, PC, and then it takes a very few percent of the PC to make this PS. So we took a genetic approach and analyzed mutant in the PSS1 gene. So we are able to find PSS1 null mutants, several alleles. They have a very strong developmental phenotype. In fact, these very small rosettes here don't really grow much more than that. So then they are not able to flower really, and they are sterile. So we always have to work with the het. But the good thing is that we have a mutant. And when we measure the amount of phosphatidylserine present in this plant, so here this is a biochemical measurement of PS, we found that PSS1 is actually not making any PS. And using lipidomic, we found that PS is absent, but most of the other phospholipids, they are not massively disturbed. But I told you that we are interested in the function of this variation of PS at the plasma membrane. And so we not only look for mutants that are not making any PS, but we design basically a whole allelic series with graded amount of plants with graded amount of PS. So for that, we use artificial microRNA against this enzyme, PSS1, and we found lines with roughly twofold less phosphatidylserine compared to the wild type. And we also overexpress PSS1, and in this line, we have roughly a twofold induction of PS accumulation in this line. So those are not huge differences, but they are quite significant. And then we started to analyze the phenotype of this whole allelic series. And one of the most prominent phenotype when we grow those seedlings on plates is that the PSS1 mutant here is completely agravitropic. So the root is growing in every direction, is not able to grow according to the gravity. So we quantify this phenotype. So for that, we grow the plant vertically. After a while, we turn them and we record how they turn through time. So here, this is a kinetic experiment where we measure how the roots are realigning according to gravity through time. So in black, you can see the wild type that is reorienting its growth according to the gravity. And here you have the PSS1 mutants that are agravitropic. The amiRNA line, they have an intermediate phenotype. They respond more slowly to the gravity stimulation. And what was quite interesting is that the overexpression phenotype, the overexpression line, the gain of function line, the PSS1 overexpression, they have a faster response to gravity. So it's kind of nice because we have the opposite phenotype between the loss and the gain of function. And for us, it was also very interesting because there was a mutant that was already described like that, and it's called ROP6. So the ROP6 loss of function mutant has a slow response to gravity, just like our amiRNA line. And the gain of function ROP6 has a faster response to gravity, just like the gain of function of PSS1. So we thought maybe PSS1 or PS production has something to do with the function of this ROP6 molecule. So what is ROP6? ROP6 stands for raw GDPase of plants. So it's a raw GDPase that can cycle in between two states, a so-called inactive or active state when they are bound to GDP or GTP. And once activated, GTP loaded, they can interact with effectors in the cytosol that will trigger usually rearrangement of cytoskeleton dynamics and orientation and intracellular trafficking. And so this cycle here is regulated by GAP and GEFs, and through usually by extracellular signal. And in the case of the root response to gravity, it was published before, it was known that auxin is one signal that is very important that can trigger the activation of the ROP6. This is unclear how this is regulated, but upon activation, then there is regulation of intracellular trafficking, microtubule dynamics that contribute to the regulation of polar auxin transport and also of the dynamics of the growth response to gravity. So we thought, okay, ROP6 and PSS1, they share similar phenotype in terms of gravitropic bending. So what about earlier readout, such as, for example, intracellular trafficking and cytoskeleton dynamics? So here we analyze many different cellular readouts. Here I'm going to just show you one, which is endocytosis that we track using the FM464 tracer. So it was shown before that actually when we express a constitutive active ROP6, so a constitutive active ROP6 is a point mutant in ROP6 that lock ROP6 in its GTP bound state. So it's always active, it's always turned on. So in this case, we have a constitutive inhibition of endocytosis, so we have less internalization of FM464. When we look in the PSS1 mutant, no PS, we have the reverse phenotype. We have a constitutive increase in endocytosis. And when we made the double mutant, what we found, the exact same increase in endocytosis as in the PSS1 single mutant, suggesting that ROP6, activated ROP6 is not able to inhibit endocytosis in the absence of PS. And it's not just that PS seems to be required for this ROP6 function, but apparently it's able to tune ROP6 function, because in the AMI-RNA line, we have kind of an intermediate response to the inhibition of endocytosis. And very interestingly, in the overexpression PSS1 line, we have a constitutive inhibition of endocytosis. And this actually mimics a constitutive active ROP6. So basically, we can mimic the phenotype induced by a constitutive active ROP6 by implants that have more PS. And this is something that for us was puzzling for a long time. And I will come back to that at the end of the talk, because we think we have at least a beginning of answer to address this. So at this point, we found that PS seems to be required for ROP6 function. So in the absence of PS, no ROP6 signaling output. And also, again, of plants that have more PS, they have a constitutive activation of the ROP6 pathway. So the question was, how does PS can control ROP6 activity? And to be honest, at the beginning, I thought this would be a trivial, question, because ROP6 is a small GTPase that is at the plasma membrane. PS is a lipid at the plasma membrane. So I thought, well, PS is going to control ROP6 targeting to the plasma membrane, and that's it. No PS, no ROP6 at the membrane, and no function. And it turned out to be a bit more complicated than that. So when we look at the ROP6 localization into the PSS1 mutant, we found no difference compared to the wild type, or something that were really minor that could not explain the absence of function. So we had to dig deeper, and we thought, what if PS is not involved in targeting ROP6 to the plasma membrane, but what if PS is involved in the very distribution of ROP6 within the plasma membrane, or the dynamic of ROP6 within this membrane? And at this point, we had to kind of stop working on PS, and we started to develop assays, microscopy assay, to actually probe this ROP6 dynamic and distribution within the plasma membrane. So I'm going to show you a few of them. The first one that we use is turf microscopy, which is a technique that creates an evanescent wave locally at the cover slip. So we can illuminate the plant cell here, but only in the vicinity of the cover slip. So it's a very shallow evanescent wave, about 100 to 200 nanometers. And so basically, all the rest of the cell is not illuminated. So there is no out of focus light. And so this is a very powerful technique with increased axial resolution. And also it's allows very, very, very fast imaging. So we can do 30 milliseconds, 50 millisecond images very easily. So you have to imagine that with this technique, we can really look specifically at basically at the plasma membrane, or what is very close to the plasma membrane. So here we look at ROP6. So you have to imagine that we are looking, making like a section through the plasma membrane. So this here, we got a very kind of homogeneous signal of ROP6. However, when you look at the constitutive active ROP6, we had a different image, because now ROP6 was found in these small domains here. These domains, they are at the plasma membrane. They are not moving. They are very stable. And we think they are like kind of a regroupment of ROP6, constitutive active ROP6, GTP-loaded ROP6. And it's not only the genetic activation of ROP6 that well, when we see this clustering, but it's also upon oxine treatment. And that's a very fast response. So when we put oxine, 5 minutes after the treatment, we can see that the wild type ROP6 now probably is getting activated, and then is getting recruited into this plasma membrane domain. So basically, upon activation, ROP6 localization is not uniform anymore. And we saw that maybe PS could be involved in these dynamics. But here, we are still limited in terms of resolution. So these domains they appear to us as a kind of a 250, 300 nanometer domains because they are diffraction limited. That's the resolution, the very resolution of our microscope. So we turn to another technique, which is a super resolution microscopy technique and also a single molecule imaging technique that's called PALM, or photoactivated localization microscopy. In this technique, we use a photoconvertible fluorescent protein called MEOs that fluoresce in green. But upon UV excitation at 4 or 5 nanometer, it's going to turn into a red fluorescent protein. And in fact, in this experiment, we only use the red channel. So at the beginning, there is no activation, so no signal. And then we will turn the UV laser, but with very low energy. So it will activate stochastically a very few of the molecules in the sample. And so these molecules, they will be very well spread in the field of view, and we can precisely localize them. And when we will localize them, what we use is a maximum energy to be able to record like single molecule and also to bleach them at the same time so that we will record their position one and only once. So here, then we can fit the Gaussian of each molecule and have a really greater resolution that is really huge because it's an improvement of resolution by a factor 10. So now we have, instead of having 250 nanometer of resolution, we have 20 nanometer of resolution. And then because these molecules, they are bleached, now we are back to the initial state and we can do the experiment again in an iterative manner and we can record the position of each individual molecule in our sample. So what we did is actually not a PALM experiment, but a variation of this, which is called SPT-PALM. And that stands for Single Particle Tracking PALM. So SPT-PALM is almost the same, but we do two small variation that actually makes the experiment completely different. The first one is that instead of using a fixed sample, we use a live sample because we are interested in protein dynamics. And the second one is that we used mild energy excitation laser, so that when we will record the position of one protein, it will not bleach immediately. And so at the next frame, we will see that it will move. And because we have a lot of precision at 20 nanometer, we can really precisely record how these proteins are moving through time. So here I show you a typical movie. So these are 20 millisecond exposure time. And each of these dots, they are single molecule of MEOs ROP6 that you can see are moving within the plane of the plasma membrane. And just to tell you that usually we do 3-minute movie, so basically 10,000 images, and we can reliably track 10,000 molecules per movie. And then we can pull all these cells together, and we have a huge statistical power on the to analyze the dynamic of this molecule. So one of the very first things that we can do is that we can retrack the trajectories of this single molecule. And you can see here they are very well dispersed. So the tracking is actually quite easy. We have very low error. So we can retrace here the trajectory of single molecules. So here in yellow, you have a trajectory of a single ROP6 molecule that we call mobile because it's kind of moving through the plasma membrane. And then we notice that there is a second population of molecules that here are in blue, and those we called immobile because, well, basically they are not moving during the time of the experiment. And that we can quantify across 10,000 of molecules. And what we found is that in the mock condition, we can fit basically this population to a single Gaussian that corresponds to mobile molecule. So almost all the molecules of ROP6 in the mock condition, they are moving at the plasma membrane. Now, when we put oxygen for 5 minutes, what we found is that now we have about 30% of the population of the molecule in the population that completely switch their dynamic behavior because they become immobile. There are those blue dots here. They become immobile, and this is a huge difference. Here we plotted the log of the diffusion. So it's a huge difference in diffusion because that's like 1 log of diffusion difference between the mock and the oxygen treated. So it's like those ROPS molecules, they are moving in the plane of the plasma membrane, and upon oxygen, clack, they become really fixed. They are not moving. And something you may have noticed here is that upon oxygen treatment, not only we have some molecules that become fixed, but it's not random, but they seem to be becoming fixed together. And in fact, if we perform clustering analysis, we can find that they are clustering together into this very small domain that we dubbed the term nanodomain because they are very small. They are about 50 nanometer in size. And we can count the number of, or estimate the number of molecules that we have in these domains, and they vary between 25 to about 30 ROP6. So they are clustered together. So they are nanodomains or nanoclusters. And of course, so we see that ROP6 is going into this cluster upon auxin treatment, upon activation. So of course, our hypothesis was that, okay, maybe once it's activated, ROP6 is getting clustered, and this is required for signaling. And perhaps PS could be required for this clustering. And so no PS, no clustering, no signaling. This could make sense. So this is what we analyzed. We cross off our MAOs ROP6 line into the PSS1 mutant, and we found that MAOs ROP6 is indeed not responding to auxin anymore and is basically staying mobile even in the presence of auxin. And so PS is required for this clustering. But it's not just that, or for this response to auxin. It's not just that it's required, but again, it looks like PS is really a regulator of the clustering, because in the PSS1 overexpression line, what we get is a constitutive clustering of ROP6, even in the absence of auxin treatment. And this is further boosted by auxin treatment. And so if you want, what we get is that even in the absence of auxin, ROP6 is clustered. And we think this is why when we have more PS, we mimic a constitutive active ROP6 phenotype, because basically once ROP6 is in this cluster, it's active and it's signaling, it's recruiting effector and it's doing its job in the cell. And so the take-home message here is that yes, small GTPase, GTP cycle is very important, but actually the precise spatiotemporal activation of the GTPase is also critical. And if even in GTP state, if it's not at the right place at the right time, there is no signal. And so is this variation of PS at the plasma membrane, is it important? We think it is, because in this cell, we have more or less PS at the plasma membrane. So we have more or less ROP6 that are going into clusters. And so we have basically a stronger response here in cells that have more PS than in cells that have less PS. So if you want, you can think about it like that. If you have a certain concentration of auxin, it will have different signaling output depending on the concentration of PS at the plasma membrane. So auxin is a regulator, but PS is also a regulator. And here, I hope I kind of demonstrated that we can link what is going on at the nanoscale all the way to the tissue and organ scale. So I think this work on lipids fit very well into this team of the plant cell atlas, because obviously lipids, they are important at the cellular scale, but I showed you that they are also important for the nano assembly of signaling molecule, for example, at the plasma membrane, but also they are very important. They are patterned also at the tissue and organ scale, and this is very important as well. So what we are trying really to do in my team is to probe the function of these lipids really across scale. And I want to thank all the people from my lab, especially Mathieu Platt, who was a very gifted PhD student that worked on this PS project, together with an engineer in my lab that developed all this microscopy technique, Vincent Bail, and all our collaborators. And I want to thank you for your attention. Thank you.
Great. Thank you, Yvon. We have about 6 minutes for questions. Start with first question in the chat from Ahmed. He's asked, do you know if the split PAM technique has a lower size limit for the protein of interest?
I'm not sure I understand the question. A lower size. Basically, the resolution that we get is about 20 to 30 nanometers. So, of course, if you want to look at structures that are below this resolution, it will be a problem. There is not many actually so far. You know, if you want to go below 20 nanometer, I guess it's electron microscopy will be the technique of choice. or you would need to couple this kind of PAM technique together with expansion microscopy. But otherwise, yes, I think that's the lower resolution of the technique. Also, you have to think that just the MEOS in itself is about 7 nanometer. So, you know, if already you have such a kind of big tag and you want to look at something smaller, this could be problematic.
Okay, great. We have second question from Benjamin Cole. Apart from ROV6, how generalizable is this mechanism for modulating diffusion of proteins in the membrane?
So I think it's, I don't know if it's going to be always PS. I think many of these lipids are partitioned within the plasma membrane. And I think basically it's going to be the rule, and the exception will be to have proteins that are not clustered. And it's very hard, actually. We even ask ourselves, do we, in the end, can we find proteins that are not clustered? And yes, we can, but it's not so common. And we find some proteins that have like kind of much faster diffusion, but the majority of proteins, especially the ones that have an extracellular domain, they don't diffuse very much and they tend to cluster.
Great. The next question is from Yvon Stahl. Is there a difference in clustering within different cell types of the root meristem?
So the only cell type that we can look at with turf is the epidermis, right? I mean, maybe the lateral root cap, but we don't really like to look at these cells because they are dying and they are weird. But otherwise, we don't really have access with turf microscopy to the cortex, or because we can look only at what is in contact with the cover slip. We are starting to use other techniques where maybe we can access deeper into the tissue, but I cannot answer right now. So what we can see more easily is the difference across the differentiation of the epidermis. And there we can see, for example, that there is this variation of PS, and this impacts the clustering of ROV6. Very likely this impacts the clustering of other small GDPAs, and for other protein, I don't know.
And next question is from Lisa David. Can the split POM be used for proteins that are mobile in the vasculature? Can it be used for peptides?
So in the vasculature, so the problem with SPT-PALM is that you are imaging single molecule, right? So you need to have a very, very sensitive imaging system. And that's why we coupled, maybe I didn't mention it, but we coupled, when we do the PALM imaging, we do it using turf because that's a very sensitive technique. So if you want to go deep, like in the vasculature, this is going to be difficult. So I guess you can do particle tracking, but maybe not single molecule particle tracking. And peptide, that's a very good question. So I think it can, providing that you can label your peptide with either photoconvertible fluorescent protein, but that's kind of big compared to peptide. So not sure this is going to really reflect the diffusion of the peptide then. But maybe it's possible to label peptide using chemical dyes that are photoactivable and to follow their diffusion. There, of course, if you are looking at peptides that are in the epidermis and especially in the outer cell wall, this would be easier because then you can use turf. But yes, I think the labeling is going to be the tricky thing.
Great. Let's thank Yvon for a great talk, and thank you for people asking questions in the chat.